Unraveling the immune-epilepsy connection, a controversial journey. The intricate relationship between our immune system and epilepsy has long been a topic of fascination and debate. While emerging research points to immune dysregulation and neuroinflammation as potential culprits, the exact mechanisms remain shrouded in mystery. This study delves into the causal effects of immune cells and inflammatory proteins on epilepsy, aiming to shed light on this complex puzzle. Methods and findings. Utilizing the largest available genome-wide association study data, researchers explored the impact of immune cell phenotypes and inflammatory proteins on epilepsy risk. Through a series of sophisticated analyses, including Mendelian randomization, (MR) methods, they uncovered a fascinating web of associations. Immune cells and epilepsy risk. The study identified 32 immune cell phenotypes linked to epilepsy risk. Some, like CD19 plus B cells, increased the risk, while others, such as regulatory T cell subsets, offered protection. Further analysis revealed distinct patterns for generalized and focal epilepsy, with 30 and 26 immune phenotypes associated, respectively. Inflammatory proteins, the controversial players. Here's where it gets controversial. 8 inflammatory proteins showed suggestive causal effects on epilepsy. CC chemokine ligand 23, CXC motif chemokine ligands 6 and 11, and vascular endothelial growth factor A were implicated in increasing epilepsy risk. On the other hand, interleukin 13, IL-13, leukemia inhibitory factor receptor, tumor necrosis factor, and osteoprotegerins seem to provide protection. But how do these proteins exert their influence? Mediation analysis, unveiling the mechanism. Mediation analysis revealed that inflammatory proteins mediate a significant portion, 6.3 to 13.5%, of the immune effects on epilepsy. For instance, CD14 plus CD16 plus monocytes increase epilepsy risk by elevating CC chemokine ligand 23 levels, while effector memory double negative, CD4-CD8, T cells reduce epilepsy risk by upregulating IL-13. These findings suggest that inflammatory proteins act as partial mediators in the immune epilepsy pathway. Sensitivity analysis, robust results. Sensitivity analyses confirmed the robustness of these findings, indicating that the associations are not mere statistical anomalies. Although none reached Bonferroni-corrected significance, the study implicates B cells, monocytes, regulatory T cells, and cytokines, like IL-13 and leukemia inhibitory factor receptor, in the pathogenesis of epilepsy. Conclusion and future directions. This study enhances our understanding of the immune inflammatory pathways in epilepsy and identifies potential therapeutic targets. However, the journey doesn't end here. Future research should validate these findings across diverse populations and delve deeper into the molecular mechanisms underlying these associations. As we continue to unravel this complex puzzle, one thing is clear: the immune system plays a pivotal role in epilepsy, and its regulation could hold a key to better treatment and management. Thoughts and comments. This study raises intriguing questions. Do you think the immune system is the primary driver of epilepsy, or are there other factors at play? Are there any specific immune-related treatments you'd like to see explored further? Share your thoughts and let's spark a discussion.